Okay. Hello, everyone. I'm Josephine Nalbantoglu. I'm at the Montreal Neurological Institute, and I am a professor at McGill University. And today I'm going to be presenting on the work that we've been doing using the BioFlux system for cell migration. So my lab works on the Coxsackie and adenovirus receptor, and this is a cell adhesion molecule of the super IgG family. And this slide shows the structure of this molecule. So it's a pretty classical one with an extracellular domain made of two Ig domains, transmembrane, and then a C-terminus that's quite short cytoplasmic tail. So as its name indicates, CAR stands for Coxsackie and adenovirus receptor because it was first identified as a high affinity receptor for these viruses. But since then, its physiological function has been discovered in many different systems, actually. So as an adhesion molecule, it's part of the tight junction. It interacts with ZO1 to establish transepithelial resistance. And it's also quite useful in development because it has been shown that knockouts are mice that have been knocked out in Coxsackie and adenovirus receptor die quite early of cardiac abnormalities. So it's been thought to be involved in functioning of the nervous system and the cardiac system and even the kidneys during development. And it also has a role in cancer, and this has come about because a lot of labs, including ours, uses adenovirus for gene transfer for cancer cells to be able to put in cytotoxic or lethal genes into these cancer cells. And what was discovered was that the adenovirus really infected these cells quite poorly because they had lost the cells themselves had lost the CAR. So since then, CAR has been thought to be a putative tumor suppressor, and it's so I'm trying to get this down. And in cancer, we can see that CAR expression is downregulated in many different cancers: gastrointestinal, bladder, prostate, ovarian, head and neck, and so on. And in all these cases, the potential role of CAR has been seen to be a tumor suppressor. And in many of these cases, it's also been shown to inhibit cell migration. So my lab is interested in glioma, and that's the model system that we're using. So we have expressed the CAR in a human glioma cell line, the U87, which is quite a classical glioma cell line, that doesn't have any CAR expression. And what we've shown is that the full-length CAR, as shown here, does inhibit glioma cell migration, cell invasion, and also tumor growth. And that the C-terminal is required for this inhibition because this shortened CAR does not have the same effect. And most of this work we did with Boyden assays. So these are cell migrations through a transwell. And what we showed early on is that the control cells that we call LNCX that have been infected with the control retrovirus have quite robust migration through the transwell, whereas the ones that express CAR, the U87 CAR, their migration rate is much lower. When CAR expression is downregulated through an antisense, what we see is that the migration rates go up, and they are basically about the same as the control cells. And we have the same type of migration when the cells are expressing the truncated CAR, which doesn't have the cytoplasmic tail. So what we've been interested in is the mechanism of how this cytoplasmic tail inhibits cell migration and invasion. And through the years, what we have used as assays in the lab are two commonly used assays to measure cell migration. And this is the transmembrane Boyden chamber assay and the scratch wound healing assay. And we basically struggled with both assays, and for different reasons. In the Boyden chamber assay, cells are placed on the upper chamber. A chemotactic agent is placed in the bottom chamber, and then the cells are allowed to migrate through, and at the end of the assay, cells are fixed, and the numbers that have migrated through are counted. So first of all, the manual counting of these cells is really tedious, and also the number of cells that migrate through is very dependent on the number of cells that are seated in the upper chamber. And depending on day-to-day assays, the gradient that's established from the bottom chamber of that chemotactic agent could be quite variable. And I think the worst thing for us was that this is an endpoint assay, so it's not dynamic. So we could have many different chambers that were seated with different conditions, and we never knew whether they were going to work or not till the 16 hours were over, everything was fixed, someone would take them to the microscope and would start counting. So in the middle of the assay, we couldn't even tweak anything. So this is an endpoint assay. The other assay that's used quite commonly is the scratch wound healing assay, in which a plate of very confluent cells is scratched, usually with a pipette tip or a needle. Unfortunately, in our case, our cells don't really make a confluent bilayer, monolayer, so that we can't have very reproducible scratches, so that the edges of the scratch are quite variable. So our monolayers are not really that amenable to this assay. As well, when you scratch, the extracellular matrix underneath the cells can be damaged, and the cells themselves are damaged, so that they will release other agents into the medium, like cytokines, that will affect the result of the migration assay. And of course, since this is in a monolayer, there's no gradient of chemotactic agents. All we're looking at is just plain migration of cells where there's no direction that's being established, no polarization of the cell is required. So that's why we went to use cell migration assays with the BioFlux system. So in this, what we do is we seed our cells in the BioFlux apparatus. We usually seed the cells on a fibronectin matrix, and they attach quite well. And once the cells are in the channels and they have attached, then we can pass medium, either the conditioned medium, which contains the chemotactic agent, or the starvation medium, which contains the DMEM 0.5% BSA. So that what you have is you have these two channels. They're uniformly seeded with the cells, but they receive two different media. We apply the shear flow, and then we image every 10 minutes. So we can see what the cells are doing under the microscope as the assay is being carried out. And this is just to show you what it looks like under the microscope, where you have our control cells, the U87LNCX on top, and the U87 CAR cells at the bottom. So the way this is arranged now is that the cells are seated uniformly, but the medium that's going to be passed is conditioned medium. The chemotactic agent is in the top half of this channel, and the low serum medium is in the bottom. And it's the same for all, so that if the cell is going to be migrating, it's going to migrate towards the chemotactic agent, towards the top. And if we play a video, which has been acquired every 10 minutes of these cells migrating towards the chemotactic agent, what you see is that the control cells are moving from the bottom towards the top. The CAR cells, there's much less movement, but just seeing this in lifetime was, when the machine was demonstrated, was quite exciting for us. But we have a lot of different mutants and a lot of different cells that we want to look at. And although this was extremely satisfying, we wanted to be able to quantify what was happening. And that's what we've devised using tools that are available on the internet. And that's what I'm going to be talking about today. So for the film that you saw previously, what we can do is just take each cell and trace its trajectory during the time of the assay. So we were imaging every 10 minutes. Later on, we were imaging every hour. And you can keep this going for many hours. What we discovered was that 7 hours was sufficient for us to get meaningful results. So that what you have here is an example of the tracking of single cells in the lower chamber after 7 hours in BioFlux, and each cell is being tracked with these different color paths. So to do this, we load the images into ImageJ software, and it's got a plugin that's for manual tracking, and we convert these images into a stack. We track each cell, and we save the results as manual tracking file. And then we import this file into a different software that's also available on the internet, and it's called Chemotaxis and Migration Tools. This software allows us to get the plots. It allows us to get the migrated distance, the angle, and so on. And we can then graph everything and get statistics with any software, the statistical software package that you want. So that we've gone from seeing how the cells migrate in the film, in the video, to getting quantitative data out of it to be able to compare between experiments. And I'm going to show you some of the representations that we can obtain this way. So this is for our control cells, and this is the migration pattern for the U87 LNCX. So this was the cells that were acquired through videos over a 7-hour period. So we had imaging every hour in the BioFlux. We analyzed it with the chemotaxis and migration tools, as I described previously. And what this tool did was it plotted trajectories. And from the trajectories, we can also get a graphic representation of the movement. So right away, you can see that we have cells. The 0 is the border between the channel that has the chemotactic agent and the channel that has the serum-free medium. And each dot represents the tracking of each cell. So you can see how far each cell has traveled, and also the angle at which it has traveled. So in this case, we have cells that travel almost 125, 130 microns. And this is a second experiment done with these same cells, and this is a third one. So these experiments were done over a course of about a month and a half, I think, and they were done by 2 different people in the lab. So this visual representation was really very appealing to us because we could kind of see what each cell was doing. And the other thing that we noticed was that there were some cells that really weren't moving towards the chemotactic agent, but they were just either staying near that border or moving in the opposite direction. And this will become important later on when I show you other results. So once we have these trajectories that are plotted, well, they can be all the data then can be used for graphic representation. And the types of things we can get from this is measurement of distance. So we can measure total distance, which is the distance traveled by the cell from the beginning to the end. Then there's total distance negative, which means that For the cells that I showed you where we had the red paths, those were cells that were traveling in the opposite direction from the chemotactic gradient. So this is when the angle is less than 180. So these values are negative. So they get subtracted so that the total distance now represents only the distance that's covered towards the chemotactic gradient. And then there's something called the Euclidean distance, which is really quite important for us because this represents the shortest distance from start to end. So you get a sense of movement because you could have a cell that's traveled seemingly a long distance, but it's just not gone in a straight line, but in circles. So this is the distance from beginning to end, so that we're really getting rid of all the cells that are just moving about in a circle. So we did all this to be able to compare our different cell lines, and I'll show you some trajectories that we've gotten with these tools. So the top are the first three that I showed you previously with the control U87 cells, and the bottom are the cells that are expressing CAR through the retrovirus transduction. So just visually looking at it, you can see that in most cases the CAR cells really travel much less. They have maybe a couple of cells that go up to about 100 micron. Most of them remain near that border between the gradient and the serum-free medium. And from all the dots and all the accumulation of these dots, you really get a visual view of how little migration has happened, and that some of it might also be not driven by the Q. So these, again, were done at different times by different people. And the other thing that I'd like to point out is that you really don't need to have that many cells, because in most cases we try to track between about 29 or like 25 to about 40 cells. That's just the right density for us to be able to follow each cell's movement during the course of the experiment. So the trajectory that we've gotten here, for example, says that 30 cells were tracked, 21 of them moved towards the gradient, 9 of them moved downwards. So when we graph these, this is what we see, that the control cells in blue go longer distances, total distance. They go longer distance even when we subtract the negative in case they're moving away from the Q. And they're also longer distances Euclidean, which means that they're going more in a straight line and not just in circles. And we've done this, the cell lines that we've been working with have been mainly retrovirus transduced cell lines, but we've done the same thing where we put the CAR in a plasmid, expression plasmid, and we did the stable transfectants in U87 cells. And when we put them into the BioFlux, they really behave the same way as the retrovirus transduced cells, so that when I'm talking about quite good reproducibility, it's also whether, depending on how we express it, whether it's adenovirus, retrovirus, or transfection, we get the same results in terms of the biological phenotype. So, well, we already knew from the Boydens that the CAR cells really were inhibited in migration compared to the control cells, but what we're interested in is trying to decipher the mechanism of how this happens. And one of the students in the lab wanted to examine the cytoplasmic tail, but specifically the 4 tyrosines that are present in this tail. These at 269, 294, 313, and 318. So all these were mutated into alanines. And I'm going to show you data on the 294 and 269. So when we transfected and made stable transfectants, pooled transfectants of the 294 cells and put them in the BioFlux, these were the patterns that we got in the trajectories. And it's quite apparent, first view, that these cells are migrating very differently from either the control cells or the CAR cells. You can see that the distance is much longer. They're going all the way up to about 200 microns. There are many, many more cells that are migrating. We see very few dots that are stuck near this origin at the border between the Q and the serum-free medium. So it can give us the distance of migration, the direction of migration. But the other thing that we found quite remarkable was that this was the first time we were seeing so many cells that were migrating away from the Q. Because as I was saying before, we get the counts. So for example, in this one, we have 28 cells that were tracked, and 14 of them migrated toward the Q, but 14 of them migrated away from the Q. So this is into the serum-free medium. And here it's the same thing with 35 cells that are tracked, 25 go towards the chemotactic agent, and 10 of them are going downwards. It's almost as though they're not polarized. And since then, we've done quite a bit of work, and we've realized that this mutation in the tyrosine at 294 to an alanine changes the phenotype of these cells so that one of the signaling pathways that we are studying, the STAT5 signaling pathway, is constitutively active in these cells, which means that they don't really need the chemotactic cue in order to start this migration. And we think that we have a visual representation of that right here, where the cells are perfectly fine to be moving in whichever direction because they're not really being cued towards the agent, because their STAT5 pathway, which is responsible for this migration, is constitutively active, so it doesn't really need the chemotactic agent. And this is something that we wouldn't have been able to see in the Boyden chamber, because we see in the Boyden it's a transwell, so it just goes through the membrane and you're just counting whatever's come through. You have no idea that something is moving away. And it's the same thing with the scratch assay, because in the scratch assay, cells move into the gap. You don't know that they're moving backwards away from that gap. So this to us was quite interesting. But the other question was, would any mutation do this? I mean, was this something that was specific to Y294, or any way that we tampered with these tyrosines would have the similar effect in the assay we were doing? So we compared it to our other mutant, which is a Y269 alanine. And these are represented here at the bottom. And what you can see here is that this is very, very different from the top pattern. So that most of these cells, for example, here, 45 cells were tracked. Most of them remain very close to this origin, to the border. So we have about 23 that move, but very little, because the longest is about 60 microns, but most of them are around 20, 25 microns. Some of them move away from it, but again, very short distance. And we've done this 3 times, and in all 3 times, pattern is very similar and extremely different from this other one. So it's not just the fact that we've mutated one tyrosine in the tail, cytoplasmic tail of CAR, that's giving a difference in the migration, but that each of these tyrosines play different roles. And we can quantitate this, and this is shown here, where we're looking at the distance traveled by the U87 CAR mutants over 7 hours. And we've done about 6 replicates of the 294A and about 3 replicates of the 269A. And you see that in each case, the 269 is migrating less. But what's really important is that we are capturing how much more the 294 mutants are traveling in this assay. And even when we subtract half the cells that are moving away from the chemotactic agent, like the red paths that we were looking at, the total distance, including the negative, is still much larger than for the 269. And the Euclidean one is also the same, that it's much longer. So that now we know that these 294 cells travel much further. And we also have evidence that they travel even longer distances than the control LNCX cells, so that this mutation really has changed the response of these cells to the chemotactic cues. and plays quite a large role in the migration of the cells, and it's one of the reasons why CAR seems to be such a good modulator of cell migration. Another thing that we've been using, and we've just started doing this with the BioFlux, is to look at the early effects of therapeutic agents. Because we're interested in glioma cells, we're always looking at different agents that interfere with different pathways in these cells. And here, the example that I'm showing you is just using an inhibitor of protein translation. So when we use these inhibitors of protein translation at very early time points, which is within about 24 hours, there is no cell death that we can detect. There is a lower proliferation compared to the vehicle. These agents come in the MSO. Compared to the vehicle, the proliferation is reduced by about 30%, but there is no cell death. And we wanted to know whether we could see something that was different also in migration in the presence of the therapeutic agents. So we treated the cells with the agent for about 12 hours, and then we watched, videotaped the migration of the cells, whether they were in the treated with the MSO or treated with 4-EGI. Again, we have a chemotactic agent, so we're looking at chemotaxis and their ability to migrate towards this chemotactic gradient. And what we see here is that obviously the MSO does affect the migratory behavior of the cells, but when we compare the vehicle to the inhibitor itself, which is shown at the bottom, that there's a difference in the migration pattern, so that we have much less migration in the cells that are treated with the inhibitor, whether it's the total distance, the total subtracting the negative, or the Euclidean distance. So that right away we're seeing something into migration by quantitating fewer than about 50 cells, that there is an effect of this inhibitor on migration. Long term, the inhibitor at around 96 hours or so does cause cell death. So that we think that we can use this for very fast, early time point screening of a lot of these inhibitors. At this point, I just wanted to show just a little bit how we use these tools, and these are just basically screenshots of our analyses. The analysis is quite straightforward. The tools are there on the internet, and what we use mainly is ImageJ. So the video that we have, the images that we have, we open them, we export them into ImageJ, and then we take all the images and we create a stack. So after they're loaded, we load images from each hour, so that for a 7-hour experiment, we will have loaded about 8 images. We load them into ImageJ. We create a stack. We convert everything so that under image, we can go to stacks and then convert images to stack. And once we do that, there are plugins in ImageJ that will do many different things. And one of the plugins is manual tracking, so that we've got one of these pictures again where we've got control cells on top, our CAR cells at the bottom, and the chemotactic agent is in the top of each channel. So we're measuring, really trying to track these cells, how they move in this gradient. And in the plugin, when we're going to be doing the manual tracking, we just add a track to each for each image, for each cell, and we have all the parameters that can be fit in here. Different people are going to have different parameters, but what we do, our time interval, for example, is 60 minutes, so that's what we enter here. Our distance for pixel is like 1 micron, so that's entered. So any parameters can be entered at this point, and then each cell is going to represent one track, and these tracks are going to be accumulated. So this is an example of where we're tracking the cells. So you can click on a cell and you follow that cell through the stack, through the 8 images, and you can overlay the dots and lines so that the data for each cell is entered into this tracking module. And this is just an example of cells that have been tracked. For example, this one started here and over the 8 images moved to here. This one started here and moved here. This is an example of a cell that moved quite a bit, but really in circles. So this is when I was talking about the Euclidean distance. When we say distance from beginning to end, it's just going to be a very tiny distance, although the overall distance might be equivalent to the same as this cell. This one is really not going anywhere and turning in circles. This is a cell that's moved quite a bit. So what you can see is that we can really track about 30 to 40 cells, and this is the right density so that you can see cell movement from one captured image to the other. And we just keep adding these cells, and then we can save the file as a manual tracking file. And the manual tracking file can then be imported into the chemotaxis and migration tool. And this is the tool that's going to really plot the trajectories based on the tracking that's been done. And for this, what we get is for each track number, we get where it started, where it ended up, and what the angle was, so that any angle that's more than 180 is going to be considered negative. And those are the red tracking paths that I was showing, where cells are moving away from the chemotaxic gradient. And then they are plotted. The trajectories are plotted, and at the same time we get the accumulated distance, we get the Euclidean distance. So we're getting this for each cell in each culture, and these can be exported to an Excel sheet or to any stats program to be able to analyze and see differences. So this is just to show that these are quite easy to use as software, and many different stacks can be gone through in one day, and it's actually taking us less time to do this than to count thousands of cells through the Boyden, because we always have to seed quite a few cells to be able to get any significant differences in the Boyden chamber. So from what we've seen so far is that this has quite real advantages for what we're doing. The best is that it's real time, it's dynamic, so that from the very beginning we can see if an experiment seems to be working. We don't have to wait for it to be ended to discover that something went wrong. Another thing that's been a pleasant surprise is that it's really reproducible across users, so that there are, at this point, 2 people in my lab who use it with different cell lines, and it seems to be, it's very reproducible. We get similar patterns, whoever's doing it over different weeks and different passages of cells. You use very small volume of reagents as you're putting when you put the shear flow on, and if you're using, for example, the inhibitors, you don't need large amounts. It's the 7-hour experiment that uses very few microliters of the reagent, and sometimes these can be quite precious. It allows us to see the migration path, not just whether they're moving, but really how they're moving and their trajectories, and that has been very, very satisfying. I think we're all visual people in my lab, so that we can hardly wait for these plots to be produced. And we find that the quantification is actually quite rapid from what we've seen so far. So this is the end of this presentation, and if there are any questions, I'd be pleased to answer them.